Yeah, what we are going to do is to talk a little bit about our MDS system. And I want to give you some advanced training about how it works and some more information. So some of you may know me already. Matthias Bold is my name. I'm a professional service manager for the EMEA region. I have some background in food technology and brewing engineering, but I spent then quite some time in the medical business as a quality manager. And in 2010, I thought, yeah, food is a good place to be. And I went back to my roots. And since 2010, now I'm working as a professional service, starting in Germany, then Austria, Switzerland, then the Benelux added, and now I have Europe. So I'm getting more and more, but that makes a really lot of fun here. What are we talking about today? Or stop, I would like to show you some of our portfolio before, because some of you may know 3M is a company doing Post-its. Yes, we do. But we are also doing Petrifilm plates since 35, 36 years. We have our pathogen platform. This is topic of today. Allergen testing is pretty new since 2 years in our portfolio. A very big part of the portfolio is sample handling. Sample handling a little bit connects everything, Petrifilm and pathogen testing, because it's a very, very important part of the whole food safety testing. Hygiene monitoring for cleanliness testing with the main product, the ATP testing, but also protein testing to cover slightly different other topics. And we have our UHT beverage testing with the MLS2 instrument for the testing for sterility of UHT or ESL products, and new is juices, and so even a little bit more complicated to test products. So this is the portfolio. But now I'm coming to the point that I started 2 times. I will give you a little bit an overview about pathogen testing, how our molecular system works. It's molecular, PCR is also molecular, but there is a slight difference between both systems. I will show you the workflow, how the workflow works, because this is really important. The workflow is really an easy one. And if you see how easy it is, then you see why this system is really a great system and provide a lot of benefits to the laboratories. We also look a little bit, but not too deep, in the global regulations. So where does this system fit? Where is it accepted? What kind of certifications do we have? So continuing, I will now switch off my camera and leave you with the slides. We will have time at the end to talk about some questions. So we have enough time. We are a small group, so it will be easy to have some discussion or questions if you have at the end. I'm starting with pathogens in general. So what are we doing here? So why are we doing pathogen testing? Pathogens are organisms that can create diseases. So the so-called foodborne diseases like salmonellose, listeriose, so sometimes really serious diseases. Therefore, these kind of pathogens should be absent in the food. We don't want to have them. And because of this, most of the testing we are doing is a qualitative testing. So we look really for a yes or no answer, and we hope to get a no answer. This is the expectation. Some foodborne pathogens require a quantitative assessment. So there is a threshold, and we know it. And if you are below this threshold, nothing will happen. The people can stand it. So it's not a risk to our health. But if this threshold is exceeded, then the risk is getting much bigger, and people can get ill and can get seriously ill. Typical foodborne pathogens, the well-known ones are Salmonella, Listeria monocytogenes, the pathogenic E. coli strains, Campylobacter is well-known, and Chronobacter. If you talk about baby food or special nutrients for sick people, then Chronobacter is very important here. Campylobacter is normally much underestimated. It's more present and you can find more of them in the food environment than Salmonella. But Campylobacter is not really regulated in the moment. They are working on that one to regulate it as the same way as Salmonella. But still, people look for it, especially in environmental testing. That's where it is pretty important. There are some other foodborne pathogens like Vibrio in fish, Shigella, Bacillus cereus, Clostridium botulinum, norovirus, but these present, compared to the others, a very low volume. So 95% at least is tested on the left side, and there's only a very limited testing on the right side. Pathogen testing is done today in the whole environment of a food factory. So raw materials are tested, end products are still tested, which is important and required by law. But there's a strong trend to look into the processing environment, and especially in the last years, we see much more pathogen testing done with environmental testing, with swabs, with sponges, with sometimes rinse water testing, because people are getting more and more aware that our processes are very complicated, very complex processes. So today, nobody is touching the food anymore, which is also a good thing. But the environment presents the biggest risk in a food factory, and therefore it's important to look also into this one. And as I said, we see the volume changing from food testing more and more to environmental testing. So we want to prevent that the pathogens entrance the food. We know that there is a 70% chance that if you find a Salmonella or a Listeria in your environment, it will end up in the food later. So it's very important to keep the environment clean and to know what's going on there. That's why we do a lot of monitoring today. Final product testing is a release criteria. It's required by law, so you have to do it. But the focus is getting more on the process and away from the final good, because at the end, if the final good is there, the whole value is already in the food, in the product. And if you now detect there's a contamination, it's too late. So you waste a lot of money and you have the risk of getting contaminated product out. So that's why the focus is getting more on the environment. A typical pathogen testing, as I said, most of the tests are qualitative. So we want to see a yes or no answer. If we do indicator testing and we look for numbers, that's rough measurement. We see the numbers, we know we don't recover all the bacteria from the surface. So it's okay, we do some monitoring. But in case of pathogen testing, we really need to know if there's one pathogen bacteria in the food. And that's why we need to do a very good sample collection. We need to take care about the kind of neutralizers we are using, the kind of sample device we are using, how we transport and treat the samples, and then we go into the laboratory. Most of the errors that happen in pathogen testing, but also in indicator testing, happen in the area of the sample collection, sample transportation, and sample preparation. That's the reason why we put a lot of focus on these things. And you may know that we just launched a new product, a new sampler, which has some benefits and really gives another level of security to this process. Then we go to the enrichment. The enrichment is sometimes underestimated by customers. Enrichment means we are looking for the one pathogen in the product. Normally there are more, but if there is one, we want to know it. And think about a soccer field. In the soccer field, huge field, covered with grass, and now somebody is putting a needle into it. You don't know where it is, and this guy asks you, no, please walk barefoot over this soccer field to detect if there is a needle. You can do it. The risk that you get the needle is quite low. Doing an enrichment means we create an environment with perfect growing conditions for the pathogen we are looking for. So this needle is multiplied many times, and we end up with billions of needles. So after the enrichment, if somebody asks you to walk over the soccer field, you will say, no, I better don't do it, because now the risk of getting one of the needles is nearly 100%. That's why we do the enrichment. And if we are not following the correct protocol, we might end up with a negative result in our detection system, but there was a positive one. And the maybe error happened in the enrichment process is causing this not correct result. And from experience, I introduced many MDS systems in the market. I did a lot of training, and I'm also involved in troubleshooting. In many cases, we found out that the enrichment process was not done correctly. Temperature may be too cold, not waiting for the time required. If you think about Salmonella, every 20 minutes they double. If you start your test 2 hours earlier, then you know how many steps of duplicating are missing, which can end up in not finding the pathogen you're looking for. So enrichment is very important. The detection part is important, but that normally works. If enrichment is okay, if sample collection is okay, so you pick up the organism, you bring them alive into the laboratory, you do a proper enrichment, then you will do a good detection. So detection is important, but many people talk about the detection process. What's happening before is very important. After the detection, we are required to do a confirmation. So is it really the one we are looking for? But that's not the only reason. These, especially molecular systems, can also pick up some dead DNA, unlikely with Salmonella, unlikely with Listeria and the other parameters, but with Chronobacter, this can happen more frequently. So you pick up some dead cells, the DNA is still there, still present. The amount of cells is high enough, so you will do a positive detection of the DNA, and then you do a confirmation if these cells that you detected are viable. Because if they are already dead, it's not a risk. So you need to check if they are viable. There are some confirmation methods you can use from MALDI-TOF, from agar method, from biochemical methods. So there's a lot of different methods you can do to confirm that the bacteria you found, the signal you got, is coming from a living bacteria that you were looking for. So this is the typical process, and you see the actual detection is only a small part of it. When we started to build or to develop the MDS system, we did a very diligent VOC, so voice of customers. 3M is a global company. We are working with many global players in the market, and we sat together with them and we said, okay, what are you wishing? What would you like to have? People said, okay, I want to have a very fast time to result. So the best thing would be to create an app on my smartphone. You make a photo of the food and the app is telling you, okay, there is no Salmonella inside. That would be perfect. But this is not what we can achieve in the moment. So we need to have a system. We need to have a whole system with enrichment steps, with detection steps. And this is what came out here. So you see a small instrument, a very small footprint. You see some tools like these blocks here for heating, cooling, for opening and closing reagents, for handling of reagents. You need a software to analyze your signal to find out if there was a positive one or negative one. You need your assay kits and your enrichment media. To tell it just in advance, the enrichment media is available. You can buy it from 3M, but we said that maybe does not make sense for every customer. So, for example, we are using a lot of buffered peptone water, and you can use every buffered peptone water. If the quality is okay, you can use it, then your enrichment will work, and you can do the detection. For the validation, we needed to provide enrichment media, but for the customer, he can decide what kind of media he wants to use. So the 3M molecular detection system, it's not a PCR system. So we are not using a thermocycler. We are not using these thermo steps of heating up and cooling down, going up to 92 degrees, cooling down again to amplify the DNA. What we are doing is a continuous process. It's called isothermal, and that's the difference to a PCR, because we are working at one defined temperature, and the temperature is 60 degrees. 60 degrees is a very good temperature for the polymerase. This is the enzyme that is doing the real job here, amplifying the DNAs. And we are using a polymerase from Bacillus stereothermophilus, which is a bacteria living at 60 degrees. So we are always in the optimum of the polymerase we are using. And then there are three elements. As I said before, there is an enrichment step. There is the detection step, or the kit we are using. Every kit has a specific color. The color can be seen later in the reagent tubes, and every color is specific for an organism. So like green is for Salmonella, yellow is for Listeria monocytogenes, and so on. The same colors can be found in the software, so you always know what you are working on. Then the third part is the instrument for the detection, for the amplification step, and the software for the detection. The process itself, amplifying DNA using a polymerase. Polymerase is happy to amplify each kind of DNA. It's not specific. But what is specific? You need a primer. The primer is the most important part of the amplification process. The primer is a specific element, so you have a primer for Salmonella, and for the other organisms, different primers. If the primer detects the DNA we are looking for, it can connect to the DNA. This is a kind of key process, because if the primer is there, the DNAs can link to the primer and the DNA string and start to do the amplification. The amplification is not different from what is happening in our body every day, millions of times, or what is happening in a PCR system, or what is happening in a further test. It's always the same process. You get a DNA, you amplify the DNA. So you get a huge amount of DNA, but this is not the detection. So amplification is one step, but that does not help. You need to have a signal to say, oh, it happens. We get a positive signal. Now we know it's a positive sample. And we are using the so-called loop-mediated isothermal amplification. There are nice videos on in YouTube to show how it works. If you Google for LAMP, then you see these. You can see these videos. It's a very robust amplification. It happens continuously. It starts slow, then it's getting into an exponential phase, so a very rapid amplification. And during this amplification, we create a lot of pyrophosphates. The pyrophosphates are the binding, the molecules that are sitting on the binding molecule that you need to amplify the DNA. So you have to remove it. The polymerase can do it. It removes the pyrophosphates, and then we have an ATP building enzyme in the reagent that picks up these pyrophosphates and creates ATP. And when we have ATP, we have another enzyme coming from the firefly, the luciferase, picking up the ATP and creating light. And now you can imagine, if you have an exponential amplification, you create a lot of pyrophosphates swimming around, picked up, built into an ATP molecule, and the luciferase is creating light. And as more ATP is created, as more light you have. And this is a direct relationship, DNA amplification and light production. So what you see in the software is a kind of real-time detection. That means there is a signal, and when this signal occurs, the software is telling you it's a positive one. A negative one is detected or not. It's interpreted by looking at the curve that it's more or less flat. There is no peak in light, and at the end of the test, the system will tell you this is negative. So positive results happen in real time. Negative results are confirmed by the end of the test. Let's have a little bit a closer look to this. So here you see a typical negative curve. It's not flat always like shown in the picture before. It can be bended a little bit like here. So there is a very slow increase, but there is no peak. So the light is getting a little bit stronger, but in a very, very smooth way. So after 60 minutes for Salmonella, Listeria would be 75 minutes. All the other tests are 60. So after 60 minutes, the system will tell you this is a negative. If you look into a positive, you see exponential an increase in light caused by the exponential DNA amplification. It's like using a dimmer, dimming the light stronger and then turning it off again. So the curve is going down after some time, and at this time, roughly, here it would be 23 minutes, the system will tell you this is a positive. This is what we mean with real time. Real-time PCR is a kind of quantitative interpretation. Let's call it this way. We are working on that one too. So this is one of the products we expect within the next time, because with all the experience we have some trigger to work on that one. In the moment it's qualitative, but you get the result when it occurs. If you do many tests, you see many curves. So here you see a row of Salmonella tests. I said green is Salmonella. You see the green squares indicating Salmonella tests. The red ones are the positives. The green minus ones are the negatives. There is a reagent control and a negative control here. Blue means they are valid. Every time you do the test, you should use a negative control and a reagent control. If they are blue, everything is okay. If there's a yellow, something happened. So now you see the curves and you don't know what is working or what is fitting to what here. So you can click on a sample place and the corresponding curve will be marked bold, and you can look at the curve. Or you can click on the error one and it will be marked also bold, and you see here a very, very low yellow curve. So what happened here? This curve, we are using a threshold. We know that each correct sample has a minimum of 10,000 relative light units. And when this is not happening, so the curve is below these 10,000 relative light units, we get an error. This error is very important because if you don't store the reagents correctly, or something happened due transport, or you're losing some reagent, or you're not pipetting something, so the pipetting was sucking air, was blocked, or you simply took the wrong well, so one well is still empty, you will get this error message. So there is no risk of having a false negative just because of such an error. It will be shown very early, and you can see what's happening. So the curves are also very important if you work with the system. You get some unexpected results. The curves gives me and my colleagues a lot of information to interpret what happened here. Another confusing picture, you see many tests. Here you see yellow circles and blue circles. Yellows are Listeria monocytogenes, blue are Listeria species. So again, you see the color indication, and you can click through the curves and look at each specific curve. You see these little waves here. This is interesting. It's not an error, but it shows us a pipetting technique. So if somebody has a very good pipetting technique, you don't see this. If somebody is not following the process correctly or not pipetting correctly, you might see these waves. So again, a very good source of information. So this is what it looks like. But there's an important process behind. The system, and you will see it later, is very easy to use. The process is also very easy to use. It takes about an hour to train somebody on the MDS system. But you should consider some other things, especially if you never worked with molecular systems before. So you have your sample preparation, working with the enrichment, pipetting the enrichment into whatever you see where we go. Then there is a sample lysis, so you need to get clean DNA. Then you start to set up your assay, and then you go into the instrument for detection. And you should always work in this direction. Sounds funny, but it's very important because you're working with DNA. And sometimes I'm joking and say, if you have a criminal mind, this is good for working with molecular systems. Because criminals should try to avoid leaving DNA somewhere so they don't get caught. And DNA is something you cannot see, you cannot smell. But if you leave it, it can be very stable. You know, sometimes criminal people are caught years after they did what they have done because of the DNA on a tool or DNA from a hair or whatever. So DNA can be very stable, and you need to avoid spreading out DNA in your laboratory because then you will get more positives than you like to. So working in one direction from clean off DNA to dirty is very important here. Never work backwards. So if you use a pipette here in the assay setup, don't use the same pipette in the sample preparation. Don't go back. Have a separate pipette here than here. Never bring tools or things backwards. Limit transfer of tools and equipment between the work areas, and I always say draw a red line. So best setup here is having this on one workbench and having this process on a second workbench. It's not like PCR where you need to have a 3-room setup. No, you can do it in one room, but it makes a lot of sense to do a kind of physical separation of these two processes. Then people know how to work, and then the risk of contamination is limited. One very important thing, if you have contamination with a molecular system, or if you want to do cleaning, cleaning only works with chloride bleach. So if you have some cleaning reagents, some sanitizers in your laboratory, they are not effective. This is also what some criminals underestimate. So they clean everything, but they are not using the correct cleaning equipment, so they leave the DNA, and DNA is absolutely stable, and chloride is the best thing to use. You can use UV light also, that helps, but if you have shadows in your laboratory, UV is also not effective. So a combination of both makes a lot of sense. Regular cleaning is very important. It's easy to do, and the system is designed for easy decontamination, so it's not a big thing. Full contamination once a month, a little bit cleaning, so cleaning the surfaces, the tools after usage is absolutely sufficient. We have different parameters. So I talked about Salmonella and the Listeria ones. We have E.colli 0157, we have Chronobacter, we have Campylobacter, and we have the latest assay is an Aztec. assay where you can differentiate between STX and EAE, E. coli. So we have different enrichment media, times, and temperatures. So there is a differentiation. In the moment, we have a Fraser, half Fraser enrichment for Listeria that will change soon. We will get a much better enrichment media that is also faster. But generally, you have to do the enrichment different by parameter. But after the enrichment step, so below the green line, the process is always the same. You pipette 20 microliter of the enrichment into the lysis tubes. The lysis tubes are ready to use. Open it, pipette into the lysis tube, done. Then you do a lysis step. So you put the rack with the lysis tubes 15 minutes on a 100 degrees heating block. That opens the cells, that cleans everything and releases the DNA for testing. Then you do a 5 minutes step for cooling at room temperature. So just pick up the block and move it to another block that is just staying around in your laboratory. Good thing is, you see the pink color here in the background. The pink color turns yellow during the heating process. And when you are back at room temperature, it's pink again. So there's a color indicator of temperature, which makes it much more convenient to do the testing to the process. After cooling down, you pipette again 20 microliter from the lysate into the reagent tubes. The reagent tubes are ready to use. There is a lyophilized pellet inside, and by pipetting the reagent into the tube, the pellet is activated, so you don't need to do any kind of reagent preparation or something, just do the pipetting. 2 pipetting steps are sufficient to run the process. And then you go into the system, and by clicking a button in the software, the machine opens. You put in the rack with the reagent tubes, and by closing the instrument, the test starts. So you don't need to do anything in addition. And this protocol is valid for all the different parameters. So if you work with different parameters, it's easy because the color coding shows you what you are doing. The color coding is visible in the software, and you only have to follow one process. So the likeliness of making mistakes here is very, very limited. And having only 2 transfer steps, pipetting step, means the risk of contamination is also limited. Every kit contains this kind of paper here showing how to run the test. Let's go a little bit through this process to show you the different steps here. If you follow this link, you can access a video in the internet showing how the process works. So first thing, you need to mix the lysis tubes. So there's a block of 96 lysis tubes. You take in your hand, you turn it once over, you turn it back, and that's it. Some smart customers, that was really something I like to see, they put the kit box in the refrigerator, they put it over the top, so they turned it one time. That means when they take it out, they have to turn it again to open it, and that so the mixing step was already done. The lysis should get to room temperature, can be overnight, so you take out the lysis tubes the day before. You can leave them for 2 hours on the workbench. You can put them for 1 hour in a 37 degrees incubator, and some customers simply put it 30 seconds on the 100 degree block, which is also a very quick step. Then you pipette out of the enrichment into this lysis tube. I said there are only 2 transfer steps, but here I recommend sometimes a third one, because going with a 20 microliter pipette into a bag with enrichment, there is a high risk that you touch the wall of the bag with your pipette, and that can cause contaminations. So the recommendation is to use a Pasteur pipette, taking out 1, 2, 3, 4, 5 milliliter. It's not important. Put it in a small tube, then pipette the 20 microliter into the lysis tube. So that's good practice here. Always use filtered pipettes. That's also in regards to contamination. And look that the pipette is calibrated routinely. Cleaning and decontamination of pipettes, I learned you have to do on your own. If you send it for calibration, there's no cleaning step. So this is some wipe them with chloride. That's sufficient from the outside. If you use filtered pipette tips, that should be always used when you are using molecular method. It's also sufficient. So after pipetting into the lysis, you go on the heating block. There's an aluminum block, and the block with the lysis tubes that you can see here fits on this block. So you don't have to do anything, just place it on the aluminum block. 15 minutes, 100 degrees, you will see the lysis turn yellow. After 5 minutes cooling at room temperature, it will be pink again, and you can do the next step. So you don't need to measure a temperature, you don't need to measure the time, just look at the color. The only temperature measurement you should do is the 100 degrees. So if you are using a heat block, you set it up to 100 degrees, it's good practice to have an external thermometer to frequently look that the 100 degrees are really reached, because this temperature is important. So what is the lysis doing? You pipette your enrichment. The enrichment contains of cells, of matrix materials, so whatever. So if you do an enrichment on meat, you know there's a lot of stuff inside. Then we have calcium ions inside that break everything, clean everything. So during the heating, the cells are opened. All the waste we don't want to have, so the matrix material, cell walls, what kind of material ever, is captured by nanomaterials. They create chelate blocks, so they move down to the bottom of the tube. The free DNA will move up to the top of the tube, and then when it is cooled down and you do your pipetting, don't go to the bottom of the lysis tube. The first third or 2/3 of the tube is okay. Then you can pipette the clean DNA and do the next step. The next step is take what you pipetted, so the lysate, and pipette it into the reagent tubes. And there are these pellets. And you see here a very, very small white ball. This is a pellet that got loose during transport. Okay, that's easy, it's not a problem. If you would lose this pellet and you don't see it, the system will give you an error message. So there's no risk of having a false negative result. And the lysis pipetting, and here we come to the point where I said in the beginning, you see the pipetting technique. I show you a little video, what should be done. So you pipette out of the lysis tubes, you go into the reagent tubes. This is a salmonella test. You push out the liquid, 20 microliters. Then you go to the bottom of the tube and you mix 3 to 5 times. You see the white liquid that indicates that all pipettes are working well. And after this mixing step, 3 to 5 times up and down, you close the reagent tube with the caps that are in the box. You can use the tool here to very easy close the reagent tubes, and then you put them into the instrument. So that was the second step. Now it's getting a little bit faster. You place the reagent tubes in a speed loader tray. This is a easy to carry tray. You put them into the right order. You can check on the monitor if the color coding is the same, then you know you did it correctly. You click the start button in the software, the instrument opens, and by adding this tray into the instrument and closing the instrument, the software immediately will start to show you the curves, and you can walk away and wait for the results. This tray is very important if you want to dispose the used test. So the test is done. You open the instrument, you take out this tray, you don't need to touch the tubes. You can dispose the tubes without touching them, because here is the risk. If there's a positive one and one tube is not really closed completely, there might be some liquid outside. Imagine these tubes are full of DNA now. So you can dispose them without touching them. That reduces the risk of DNA contamination. The system itself, it's easy built. We only need a heating system for 60 degrees. We need the detectors for the light. You can see them here on the bottom of this white plate with all the black holes. There are the light detectors. This plate is a glass plate and sealed, so you can wipe with a chloride solution the inside of the instrument for decontamination. This is the heating unit for 60 degrees. It's fixed with 3 screws. It's only a quarter turn, then the screw jumps up and you can take it out. Put it into a chloride bath, rinse it with water, let it dry, put it back into the machine, and decontamination is done. So very, very easy to do. Every startup, whenever you start the system, there's a diagnostic check, checking that everything is okay. The system is okay for running. The diodes, the light measurement is working well. The heating is working well. So there's also quite some safety built into the system. And it is such a light system, you can pick it up and carry it around. Why should you do it? Why should you carry a molecular system around? To clean the place underneath. So you can take it away, you can clean it, you can place it back. So it's a very easy maintenance and decontamination. The software is, every manufacturer says our software is easy to use. I say the same, it's easy to use. You see here the color coding a little bit. Here you see the curves. You can attach one system to the computer, but you can attach up to 4 systems to the computer. We now have some customers with 3 systems. That's what I like. So they do a lot of tests every day, and it's very easy to work with the system. If you get a new system, maybe your system is going for calibration, you get a spare instrument, you only need to plug and play. So plug it into the machine, start it up, it will be recognized, the diagnostic check will be done. There will be documented the serial number of the system which each tests, so there's always full traceability, which system was used where. There's a LIMS compatibility, so you can push your data into LIMS just by clicking a button. Here are the different assays that we have in the moment. So you see Salmonella, Listeria mono, E. coli, Chronobacter, Campylobacter, Aztec. You see a gray Vibrio. I guess that will be the next one because I already got the translation of the IFU. So I guess that will be the next test released. Now we go into the smaller volume tests because some customers really want to have it because the system is so easy to use. Lysis tubes, software. Here are the reagent tubes on the bottom with the different colors and the instrument. This is the full system. This system is used all over the world. I learned last week that we are in more than 60 countries present today, but that also means 60 countries all over the world, there are different requirements. So if we talk about Europe, it's very important to have a certification or validation according to the 16140, ISO 16140, validation of microbiological methods. And yes, we have them. We are going for every test. Now we are validating the Aztec test in the moment. That will be the next one with a validation. And we always ensure that the validation is done and repeated every 4 years. If you look into the US or into UK, or if you export products into the US or UK, you might be required to use AOAC certification. So we also have AOAC. This is the US version of the validation. It's called an approval process is pretty much the same, with one difference. In Europe, we have to revalidate every 4 years. In the US, you don't have to do this. Australia, Brazil, Canada, and several other countries are accepting this system, so it can be used all over the world. But that also means for us, these revalidation processes mean they are again and again checked if the system is working correctly. It's for us a very good quality control. The most important approval for us was happened some years ago, the first time. Now it's repeated every year. The USDA, which is the authority in the US doing the all the kind of food testing, checked our system, and they needed 3 years to do it. And after this, they say this is the recommended system for environmental, not environmental, it's for Listeria mono testing, for Salmonella testing. Now they added the Campylobacter. So we are accepted as a preferred solution from, I would say, the most critical authority worldwide. Behind the USDA is the FDA. So USDA is working for FDA approvals. We offer, when a customer is starting with the MDS, we offer documents for training. We offer training in a personal way. So I'm doing a lot of trainings with customers, and we have a procedure to show the importance of pipetting quality. So you can train how to do it, and you see what different pipetting techniques can create. It's not about safety. The test will provide you a safe result, but you see a little bit here in the curves. These look very, very nice, and these curves are a little bit more nervous. So there's more variance strain coming from the pipetting technique. The system complete gives you a lot of process control. It has a very easy protocol, easy to use, no reagent preparation. So we have ready to use reagents. We have all validations available. In regards to productivity, one of the last slides will show you the speed of the instrument, and transfer steps, only 2 very limited transfer steps. So very low risk of contamination. And here you see some comparison with other systems. It's a Salmonella assay, and you see the total time is much faster. The working time is much faster compared with other systems. We did this study, and I showed this to many customers. I showed it to Vitus customers, to Bugs customers, and also IQ checked just recently, and the customers confirmed. They first said, okay, everybody can do something like this, but they said at the end, yes, you're right. This system is very, very easy to use and quick. You can get some more information if you go to this webpage here. You can get a lot of documentation, what you need. If you need anything more, you can contact LED Techno. You can contact me. I'm giving technical information, nothing more, but that's can be quite a lot. So please contact us and ask for information. It's also possible to do a demo trial, so you can see the instrument, you can see it working in your lab, and you can even have it for a short time, so that you can do your own testing without somebody standing beside you, so you can assess how the system works. Yeah, with that, I'm at the end of my presentation. I thank you for listening. I hope you are still there. I also thank LED Techno for organizing this webinar here. It's always a pleasure to work with you and to talk about our products. So if you have any questions, we still have quite some time to talk, and I'm ready to talk to you.
If you have questions, just raise your hand or you can take the mic.
Hello. How much does this cost, more or less?
Pardon?
I think this is a question somebody from LED should answer. Miki, can you answer probably? Okay, Elma, it appears nobody knows the price by heart. I have I have your contact details. The account manager will contact you and see what you need. Also because there are quite different tests, so he will come to you with an answer on that. Is that okay for you?
Yes, thanks.
Anybody does have other questions? Christina, go ahead.
Thank you. First of all, I would like to thank to Matthias for this presentation. I think it's the first time, and with my experience, that I'll see that they are really in 3M is going to the point of many issues that we find in the in the labs. I have one question, and it's about the confirmation of the results. Matthias, what do you recommend, and what will be the best way to proceed when we find a positive curve in our results?
Mm-hmm. Yeah, there are some confirmation methods depending on the parameter that can be that are provided by ISO standard. So some ISO methods. If you really look for a living organism, then cultural method is interesting. If you want to do some more identification, what strain it is, Malditoff is an option. So there are many different ways of confirmation steps, official ones and most used ones, let's say it this way. One important thing to know, if you work with, for example, Salmonella, and you don't have a pathogen lab, a pathogen lab is required when you do an enrichment of living bacteria that you can identify. And with our system, there is no such a step. So we have quite some customers without pathogen labs doing the testing with the MDS. And if they have a positive result, they send it to a contract lab for confirmation. So this is also possible and well accepted. So this is a way. I know some meat producers, they don't have pathogen labs, but use the system to do the monitoring of environmental samples or to really have more insight about the quality. Mm-hmm.
Thank you. That's very interesting. Thanks.
Mm-hmm. Welcome.
Thank you, Christina, for asking this question. If anybody else has a question, it's your time now.
Me again, sorry. So you mentioned that there are some clients that don't have a pathogen lab, but you show in one of the slides that there needs to be an enrichment period. For example, for dairy product, there was like 40 hours in there.
Yeah.
And there is no need of a pathogen lab. How does that work? Can you help me understand, please?
Yes, absolutely. If you work with Salmonella, it does not work with Listeria. But if you test Salmonella, you are only doing an enrichment with buffered peptone water. That means there is no selective enrichment step. And this is accepted. So if you do EB testing, it's the same. You also do an enrichment of colonies, but you cannot identify if it is Salmonella or any other kind of EB. And this is allowed. So after this enrichment, you kill all living bacteria by the lysis step, and then you do your detection and you might get a positive signal. What you're not allowed to do is now put it on a selective agar plate to do the confirmation, because this is the selective enrichment step. And the selective enrichment step defines if you need a pathogen lab or not. Yeah. So I have many customers doing it without pathogen lab.
And you said it only works for Salmonella and not for Listeria?
Yeah, Listeria is as long as we are using Fraser Demi-Fraser broth, this is a kind of selective enrichment. You could say that the same is used for Listeria species, but I talked to some authorities and they said, oh no, Listeria we don't accept. Salmonella is not a problem, but Listeria, we see it as a selective enrichment.
And then,
yeah.
Sorry, what do you do with the leftover of your enrichment media? So immediately to the autoclave or something?
Yes, like every enriched sample needs to be autoclaved, yeah.
Thank you.
Yeah, you're welcome. Maybe one word on autoclaving, because I forgot to mention it. These reagent tubes should never, ever be autoclaved. DNA is not destroyed by autoclaving, but the tubes are. And if you open the autoclave and there's steam coming out, you may contaminate your laboratory. Well known by people using PCR, but I know microbiologists tend to put everything into the autoclave, not with molecular tests.
Thank you, Matt. That was a very helpful tip. Does anybody else have some questions? I don't see questions coming up, Matthias. I will thank you for the webinar. It's very interesting. This webinar is also recorded and we will forward to the participants. Thank you all and have a nice day.
Dominic, thank you very much. All participants, thank you and have a great rest of the week. Bye-bye.
Bye-bye.
Thank you. Bye-bye.